This video covers section 10.2, and we're going to continue looking at the circulatory system. And in particular, we're going to look at the composition of blood. And blood, as you probably know, is a liquid connective tissue, and it has many functions, which are listed for you here. If you look at the composition of blood, it can be observed if you take a blood sample and you put it into a centrifuge. When you do and you spin it down, and that's the term that's used for when you put something into a centrifuge, then you spin it down, the more dense materials will end up at the bottom of the test tube, and the less dense materials will um, be sort of graduated toward the top of the test tube. So when you spin blood down like this, then the upper layer will be comprised of a plasma, which is about 55% of whole blood. And then you get a layer in between of these formed elements, usually leukocytes and platelets. We will look a little bit further. Leukocytes just simply referring to white blood cells. And then the lower, approximately 45%, are the red blood cells. So plasma itself is the liquid part of blood, and it contains a variety of inorganic and organic substances that are dissolved or suspended in water. And you can see um, quite a list here. So water itself actually is about 90 to 92% of plasma. Then you have plasma proteins, which are about 7 to 8%. And we'll look a little bit more at the detail of plasma proteins. We do have salts in the blood, gases, oxygen and carbon dioxide, nutrients, lipids, glucose and amino acids for energy for cells. We also have the nitrogenous waste, which are byproducts of the breakdown of proteins, and that consists of urea and uric acid, which is excreted by the kidneys. And then we have some hormones and vitamins, etc., also contained within the blood. So the plasma proteins then, the types of proteins are bilirubin and lipoproteins. They also transport cholesterol throughout the blood. You have the blood clotting protein fibrinogen, also antibodies to help fight infection. And then for maintaining blood volume, um, plasma proteins are actually too large to leave the capillaries, and so they remain within the capillaries. They don't actually diffuse out. So blood in capillaries, it has a higher solute concentrate than tissue fluid, and that actually is what causes water to be drawn into the capillaries. So the red blood cells, and very tiny, and you're probably all familiar with the shape and the structure of these biconcave sort of disc. And they're manufactured in the red bone marrow within the skull, the ribs, vertebrae, and the ends of long bones. And obviously they transport oxygen and carbon dioxide within the blood. And you can see about how many that you will find in just a cubic millimeter. You can have up to 4 to 6 million red blood cells just within a tiny little cubic millimeter. Mature red blood cells, they do not have a nucleus, and they're kind of biconcave in shape. They only last about 120 days, and then they're taken out of circulation by the liver or the spleen. It's not the biconcave shape. It helps to increase flexibility for squeezing through the capillary beds and perhaps increases surface area a little bit as compared with a spherical kind of shape. They, they contain a hemoglobin, which, as you know, is the respiratory pigment that carries oxygen. And you can see sort of the images here. The first one would be that would be a light microscope image of blood cells within a capillary bed. And then further, that would be a scanning electron micrograph of red blood cells. And then you can see the actual shape of hemoglobin, which is illustrated on the right there. Now, some of the conditions of the blood with regard to the red blood cells, one of which is anemia, and this is a common blood disorder. It results in a really tired and run down feeling. And we probably all kind of feel that way from time to time, but this is rather prolonged, and so it can have three basic causes: either a decreased production of red blood cells, or loss of red blood cells from the body. For example, some kind of cancerous conditions of the colon, let's say, where it's causing some bleeding out through the digestive tract. That can cause a abnormally high loss of red blood cells or destruction of red blood cells within the body. But most are of the iron deficiency type, so that means then that the body cannot produce enough red blood cells because it doesn't have enough iron in order to produce the hemoglobin. So generally just simply treated with iron supplements, and that generally will correct the situation. So then we move on to the white blood cells, and you can see there are various types of these, also referred to as leukocytes. They are larger than red blood cells and have a much more sort of distinctive sort of appearance. And they do have a nucleus as opposed to the red blood cells, which do not, and they appear more translucent without stain. So they're not that easy to observe under a light microscope, especially without having some kind of a stain on them. So usually if you see images of these, then they will be treated with a particular kind of stain so that they can be visualized under the light microscope. So here would be a small section of blood, a blood sample, magnified 250 times under a light microscope, and you can see the different kinds of leukocytes. You can also see some platelets there. So we have neutrophils, there's a lymphocyte, a basophil, a monocyte, and an eosinophil. Then the leukocytes, or white blood cells, they're divided into two types. We have the granular type; those would be the neutrophils, eosinophils, and basophils. And then we have the agranular leukocytes, the monocytes, and the lymphocytes. And they're categorized this way just simply upon their appearance under the light microscope when they've been stained. So the granular ones, you can see they have kind of a granular sort of appearance. The neutrophils, these phagocytize pathogens, which means they engulf them, as do eosinophils. The basophils, they release histamine to promote blood flow to injured tissues. So those are sort of the basic functions of these particular ones. Then we have the agranular types, and you can see that they don't have such a granular sort of appearance when you observe them under a light microscope. And these particular images, they would have been stained with what's referred to as a Wright's stain. And the monocyte is the largest white blood cell. and differentiates into phagocytic dendritic cells and macrophages. And then we have the lymphocytes, then we have the B lymphocytes, which produce antibodies, and we have the T lymphocytes, and we have the helper T cells, which regulate the responses of other cells, and we have the cytotoxic T cells, which kill other cells. So as far as their role in combating disease, um, the number of white blood cells either increases or decreases beyond normal. So we have normal levels of these in the bloodstream at any given time. However, will increase the number of neutrophils in response to bacterial infections. The B cells will increase in response to infectious mononucleosis, or commonly known as mono. T cells, a low number of T cells indicates if a person is infected with HIV, and it's leading to AIDS. And then a large number of abnormal white blood cells is a characteristic of leukemia. So here's an illustration of a macrophage, which is engulfing bacteria. So this would be a monocyte-derived macrophage, and they're kind of the body's scavengers. And so they engulf and engulf microbes and debris in the body fluids, such as illustrated here. Then we have platelets and blood clotting. Platelets are also referred to as thrombocytes, and um, these appear from fragmentation of large cells called megakaryocytes in the red bone marrow. So also produced in the bone marrow, and they begin as megakaryocytes, and then the larger megakaryocytes just simply fragment and become the platelets, which circulate throughout the circulatory system. Platelets, they're involved in blood clotting, sometimes referred to as coagulation. And there are at least 12 clotting factors in the blood that help platelets in the formation of blood clots. And a couple of these are prothrombin and fibrinogen. And you can see platelets are a fair bit smaller than most of the other cells. They're not actually once again, they're not actually a cell. They're just simply fragments of what was once a cell. So here, this is illustrating the clotting process. So when you have a blood vessel that's punctured, then platelets will clump at the site of damage and form a plug to partially seal the leak. And so you can see in the beside number 2 there, the platelets, they congregate to form a plug at the site of the puncture. And so then the platelets and the damaged tissue itself actually releases a prothrombin activator, which converts prothrombin, which is a clotting factor, to thrombin in the presence of calcium ions. And the thrombin then acts as an enzyme on fibrinogen to form fibrin threads. So you've got kind of a sequence of events there resulting in fibrin threads, which act to seal punctures. The fibrin threads then, they wind around the platelet plug and they trap red blood cells to form the framework of the clot. And then when blood vessel repair is initiated, plasmin, which is an enzyme, destroys the fibrin framework and restores the fluidity of the plasma. So if blood is allowed to clot in a test tube, a yellowish material called serum develops above the clotted material.